Hello everyone, my name is Elvira Piqueras Ricote and I am currently doing my master's degree at the University of Bristol. Today I will be presenting my master's project on the fossil Eolarva kuanchuanpuensis. Firstly, I wanted to briefly mention some of the reasons why Cambrian fossils are significant. As some of you may know, all modern body plants were established during the Cambrian explosion event. Studying early Cambrian fossils, therefore, gives us an opportunity to analyze the patterns of development from the ignition of the explosion all the way to modern representatives. This, in turn, allows us to expand on our understanding of the event and why we see such a rapid increase in diversity and disparity during this geological period. Early Cambrian fossils also offer a chance to look into the diacurrent Cambrian biotic transition, and are useful in exploring the different ways in which this event may have taken place. Fossils provide a unique way to look at anatomical transitions, because even though genomic studies are useful in attempting to recreate metazoan ancestors, no genomic study really could have predicted the existence of fossils such as Dickinsonia, due to its distinctive nature. Cambrian fossils are direct evidence of what was alive during this time, and can provide answers to long debated questions of animal origins and evolution. An example of one such question is whether the earliest metazoan ancestor was a larval form or an adult form. Firstly, in the animal kingdom, there are two modes of development, direct and indirect development. Animals that undergo direct development develop from an embryo straight into an adult form. In contrast, indirect development involves at least one additional step between embryo and adults, what is known as a larval stage. Larvae have a different body plan, appearance, and size to adults, as well as different food requirements and modes of life. But what came first, a larvae or an adult? You can think of the chicken and the egg dilemma as a useful analogy for this. There are two main hypotheses which dominate the long history of this debate. The first hypothesis is called the terminal addition hypothesis. This hypothesis states that the earliest metazoan was a larva, and that adult stages were added at a later evolutionary stage, either a single time or multiple times, that is, at different times for different taxa. In contrast, the intercalation theory suggests that the earliest metazoan was an adult and that a larval stage was intercalated into the life cycle a single time for all indirect developers or multiple times, again at different times for different taxa. The fossil Eolarva kuanchuanpuensis has been invoked as a fossil which could shed light on this matter. So I just want to give you a little background information about Eolarva first. So Eolarva has been recovered from the Shizhonggu section of the Kuanchuanpu Formation, which is located in the southern Chinese province of Shanxi. The formation has been dated at approximately 535 million years old, and so it represents a very early Cambrian formation. If you consider that the beginning of the Cambrian period has been dated at 541 million years. The only study published to date suggests Eolarva confirms the terminal addition theory. They categorize Eolarva as a non-feeding larva because of the absence of any apparent feeding structures such as a mouth. However, only the external morphology of Eolarva has been described so far. The study also suggests it is an cnidarian-grade animal due to similarities it shares with cnidarians such as Diploplasty. However, differences such as the apparent bilateral symmetry of Eolarva instead of an cnidarian radial symmetry make uncovering Eolarva's affinities a little bit complicated, but not phylogenetic analysis was carried out either. My study's primary objectives, therefore, are to describe not only the external morphology of Eolarva, but also the internal anatomy, to carry out phylogenetic analysis to uncover its affinities, and therefore to assess its evolutionary significance. For my first objective, I used synchrotron X-ray tomographic microscopy scan data, and the scanning was carried out at the Swiss Light Source, Paul Scherrer Institute in Switzerland. The scans were then processed using Aviso 9.7 light software, where the resulting 3D models and internal slices were analyzed. The resulting scans show an organism of approximately 500 micrometers in length. The body has a corrugated texture with longitudinal ridges extending through the whole length of the body, as you can see in figure A, and they converge at the top to form a narrow invagination or apical furrow, as you can see in yellow in figure C. There's also two elliptical structures which are positioned at either side of the body within the upper portion, which give Eolarva its strong bilateral symmetry. Each structure consists of a circular base from which spike-like structures emerge, as you can see in figure B. The number of these spikes varies across specimens, with most specimens showing 3 spikes, but some of them showing only 1 spike. This doesn't seem to be a preservation issue such as breakage, because in specimens with 3 spikes, you get 2 spikes at the top of the structure and 1 at the bottom. But in specimens with only 1 spike, the spike emerges from the center of the structure. When looking at the internal slices, we find a sac-like structure preserved at the different phase of mineralization, which in this slide is highlighted in orange. The shape of the structure is regular across some of the specimens, and it suggests that it may represent a biological structure. Furthermore, within the structure, we don't find the patterns of regular crystal arrangement, which are usually indicative of later diagenetic infillment of a hollow cavity. Therefore, it may be preserving original tissue. It is also continuous to the narrow invagination, which we described earlier in all specimens, and it may therefore represent some sort of gastric cavity as the organism fed through the invagination at the apicalmost part of the body. For my second objective, that is to uncover the phylogenetic affinities of Eolarva, we augmented the metazoan data matrix from Duan et al. 2017, which was previously composed of 47 metazoan taxa, 48 including Eolarva. I then carried out Bayesian inference analysis using the Markov K model. The analysis resolved Eolarva within a polytomy containing Cnidarian taxonomic groups. It is therefore hard to say at this stage whether Eolarva represents a stem or a crown Cnidarian. I would like to also mention that, as you can see, I didn't include some of the groups such as Condylarida or Olivoides within Cnidaria, even though they were resolved as such, because there's still a lot of debate as to the affinities of these groups, and so they're slightly problematic, and so I didn't include them within the Cnidaria green box, as you can see. Because we find a gastric cavity-like structure in Eolarva, which continues to an opening at its apicalmost part of the body, we can conclude that Eolarva doesn't seem to be a non-feeding larva, and therefore it does not support the terminal addition theory, which states that a larva was the first ever metazoan form. In terms of its phylogenetic affinities, at this stage, we can only conclude that Eolarva may represent a potential stem or crown Cnidarian, but its evolutionary significance cannot be fully realized just yet. Thank you very much for listening to my presentation, and I will gladly answer any questions you may have about my research during the Q&A session.